All right, so today I'm going to talk about how renewable bios is unleashing the true potential of pluripotent stem cells. So a little bit about renewable bio. So we've been founded about a year and a half ago based on technology from Jacob Hanna's lab. So we got the IP from Weizmann, a combination of 3 breakthrough technologies that I'm going to expand during this talk. And we're mostly interested in cell replacement therapy, rejuvenation, aging, fertility. So what we have is a stemroid models that I'm going to elaborate that enables spontaneous and holistic differentiation of pluripotent stem cells and resulting with really authentic cells. So our mission is to provide patients and different technologies high quality cells for transplantations and also for rejuvenation of cellular and all kinds of different therapies. So there is unfulfilled promise of stem cell therapies for, and the audience here knows the different capabilities of pluripotent stem cells and what they can differentiate and what kinds of therapies they can provide. However, unfortunately, there are a lot of different, unfortunately, there are only a few approved therapies to date, despite having over 30 years of research and over 5,000, it's probably even higher today, of clinical trials. And the reason for that is in vitro differentiation is, and I think I'm kind of echoing what Yaki said before, that differentiation is very complex and it's very hard to do it in vitro. And I think that when we're doing in vitro differentiation, we're trying to push the fate of the cells into a different, really certain direction, and we, a lot of the times, fail. And we're adding cytokines and morphogens and transcription factors, and eventually after a long period of time, we're this process is resulting with non-authentic cells, and these cells are not usable. And the problem is not with the pluripotent stem cells, because if we take the pluripotent stem cells, we put them into an embryo, we create a chimera and the cells differentiate very nicely. It doesn't matter if you're using mouse cells or even you're putting human cells in mouse embryos and human cells in pig embryos. The cells can differentiate, so the potential is there, it's just unleashed. So today I'm going to talk about how we're basically bridging this gap of availability of stem cells and the authenticity of stem cells. So we have a combination of 3 breakthrough technologies. One is basically culturing the cells in very unique naive media, growing embryos or embryo models outside of the uterus up to a specific point, and then producing stemroids, as we like to call them, but there are stem cell-derived embryo models. They're not embryos, they're stem cell-derived embryo models. So I'll start from the very beginning, sperm, oocyte, fertilization, create a zygote, 2-cell, 4-cell, so on and so forth, mobile blastocyst. But then, so we know very well how to grow embryos up to this point, whether different animals, humans, but there is a barrier starting after the post-implantation. So up to the pre-implantation, we know very well how to grow these, but after that, there's an issue. So we were able to grow blastocysts, derive them. Yamanaka in 2006 were able to take somatic cells and reprogram the somatic cells back into pluripotent stem cells, but they remain in primed state, and primed state is very limited and it's very committed into the specific fate, especially if you're doing reprogramming. Their epigenetic landscape kind of remains, and you need to reset that. And how do you do that? You kind of culture that with naive conditions. That's one of the breakthrough technologies from Jacob Hanna's lab. The first was publication in 2013, but later on there were more developments. The second thing was, how do we cross this barrier? So this was resolved with the ex-uterine device. And then later on, something that was very interesting is, how do we make synthetic embryos or embryo models or stembroids, as I'll refer them from now on, how do we make those just from cells alone? So no oocyte, no sperm, no fertilization, and the interesting part here is we don't go through the pre-implantation stage. We go straight into post-implantation. And that's why these are not embryos. We can never put these stembroids into a surrogate mother because they won't implant. It's not possible, and of course we can't grow them to the whole gestation of humans. So this is a video of the capabilities of the ex utero device, and what happens here is basically we took embryos at mouse embryos at day 5.5 or 6.5, we grew them in this device right here. And what you can see here is the primitive string elongating and the somites created. And if you look further, you can see this is the point where we took them out of the uterus. So these were naturally conceived embryos, and they were put in this device, and they're of course growing, and you can see all of the elongation here of the primitive streak, and the somites would be here, and all of the formation of the heart and head and everything else. And if you remove the yolk sac, you can see better images, and this is how it looks. Formation, organogenesis, everything is happening, which was really exciting. So this is mouse, and this is naturally conceived embryo, so you're probably thinking, okay, this is good, but how are you going to use this? So the next thing that we did, there was actually a stage in between that we used mouse cells to create these stembroids. And actually, interestingly enough, the human cells, creating stembroids with human was a little bit easier because we didn't need exogenous factors to create the other cell types. So what we start with is naive pluripotent stem cells, but we induce them into two other, or maybe actually three other cell types. One is the hypoblast and the extraembryonic mesoderm, which would later form the yolk sac and umbilical cord, and the third one is the trophoblast, which would later form the placenta, and so on and so forth. And how does this work? We take these 3 or 4 cell types, we put them in a microwell of aggregation plate in a certain dilution, a certain number, and what you can see here is that day 0, they just look like cells. Day 1, they start to clump. Day 2, they start forming a ball. And by day 3, and you kind of bear with me and work your imagination, you can see that the epiblast is forming, the hypoblast is forming. Day 4, day 5, you can see the yolk sac or the primitive yolk sac and the chorion cavity. And day 6, day 7, day 8, they start and grow. And if you don't believe me, you can see here the staining. And what we've stained here is for, so you can see the DAPI and the white, that basically stain all of the nucleic acids. OCT4 is a specific stain for epiblast, SOC17 is for yolk sac, and CK7 is for trophoblast. As you can see here, day 4 of the protocol, at the lower panel, you can see a textbook images. Day 4 of the protocol is highly resemblant to 9 or 10 days post-fertilization, and you can see the epiblast here and the yolk sac forming, and also the yolk sac cavity starting to form here. And as we move to day 6, resembling 12 days post-fertilization, you can also start to see the chorion cavity forming and the primitive yolk sac expanding even more. And the cool part here is that we start to see lacunas. These are the structures that secrete the HCG. Later on day 8 of the protocol, we can see the 13, 14 resemblance, and basically all of the different structures. So that was obviously very, very, very exciting. And if we look further into this, we kind of took a home pregnancy test, and we wanted to see if they actually secrete HCG, and they do. And I think that was very cool. But if we look further into them, you can see the really unique structures of our model, because we highly believe that the structure really governs the differentiation. And what we see here is the bilateral disc, which is very important for gastrulation, later organogenesis, and so on and so forth. But this structural development is important and unique in our differentiation method. And that's something that is very important to emphasize. So this, of course, made a lot of noise, and embryo models, and got the cover of Nature and Cell, and also some public press. And this is actually a non-published data that we got about a week ago. So I showed you day 13, 14 post-fertilization, and now we're talking about 19 post-fertilization. So we're moving on, and we can see the primitive streak and all that, and these structures are really interesting, because what you see here is this is the virtual human embryo atlas, and this atlas is basically from miscarriages. And what they did, they took the miscarriages and basically took them and made histology cuts and sections. So what you can see here is a actual embryo that was collected from miscarriage, and you can see here the connective stalk and the structure of the placenta here, the hypoblast here, and this is the actual embryo proper. This is the epiblast, and here you can see the amnion, and this is a stemroid, and you can see the disc here, the amnion here. This is the R placenta, and this is the yolk sac. So what we can do with this is, our vision is basically to take a skin biopsy from a patient and reprogram his somatic cells, whether it's blood cells or fibroblasts, and use non-integrative pluripotent reprogramming factors, the Yamanaka factors, grow them in primed, stabilize the conditions, then transition them into naive, stem them into the hypoblast and trophoblast induction that we're doing, to collect D3 cells and put them in the aggregation plate that we're doing, and then we're basically creating the stemroid by aggregation, mature them in our culture, ex utero culture device, up to the certain point that we need the certain cells for this patient, and then we can isolate the cells and put them back in the patient, and then you can have an autologous transplantation, no GVHD whatsoever. So that's our vision. That's what we're going to do. We're progressing, and we are isolating cells as we speak, endothelial cells, hematopoietic cells, because we can grow now. We're still characterizing this, but we can grow up to day 28 post-fertilization. So we're really excited about what we're doing and what we're developing out of this, and hopefully by next year I could show you some in vivo functional data. And I think the really special thing about our system is it's not unilateral. What do I mean by that? So when in vitro differentiation, you get one cell type, 3 cell types most. With our platform, we're growing our stemroids, and we're getting more and more different cells that we didn't even think about. So we're growing, and we can see yolk sac cells, we can see endothelial cells. Now we're starting to see HSC cells, when we're actually seeing really specific endoderm cells that would later form hepatoblast and pancreatic cells. And later on, we're planning on getting oocytes and fertility cells, and of course, beta-secreting, insulin-secreting cells, beta cells. But we're really excited about the future. This is our team. And with that, I would like to thank you, and thanks for staying so long.
In what type of extracellular matrix would you put the cells in this agri-well?
No extracellular matrix. No, but that's a really interesting question, because part of our conversation from last night, we are trying different extracellular matrices to see if it improves the process.
Because we heard that the interstitial fluid is very important for the creation of the lumen.
Yep. And polarity and all that kind of stuff, yes, for sure. And
you get it just by putting them in a medium and agri-well.
yes, however, we do believe that the axis would be, so gastrulation is really governed by the axis. So that's what we need endometrial cells to see if they're working well or not. So yeah, that's an interesting question, and we're testing it.
And until which day are you going to
continue this differentiation process?
So
Until which type of cells?
So the short answer is I don't know, but the long answer is, so we're not aiming to go past 140 days. But we know that we'll get to a certain point that now we're discovering more and more cells, so we can end up just cutting it at 100 days. But we are aiming to getting oocytes and stuff like that, that we know that usually you can get them at 70 days or later. But that's the thing about human development. So we know that we have an atlas of sections of H&E, but we know nothing about this process. And this is a really, really important process. And we know nothing about it since day 7, basically, since we implant the embryo up to day 100, we know very little. And we have very few references for different time points. So we have one embryo 16 to 19, we have one embryo 20 to 25, we have one embryo, and that's like the single cell resolution. But we know nothing about human embryo. We know nothing about human development at these time points. So I hope that we will also be able to kind of shed some light on these processes.
And can you comment a little bit about the ethical aspect of
Yeah,
this?
so we are we're head front on the ethical, and we're starting a committee about this, and the ISSCR is actually very active about embryo models today, because I think the overall notion is that these are not embryos. This is not something, and even in the UK, there is the 14-day rule, and for embryo models, it's not covering it. So you shouldn't consider it as embryos, because of course they cannot make a human. However, they can make really, really good cells from transplantation. And as Jackie mentioned before, I'm also a great believer in cells, and I think that's something that we should promote, because there are so many conditions that we can cure with really decent cells for cell therapy that I think the ethical ideas need to change in this notion, and they will change, and you already see the trend that it is changing.
So you avoid saying organ. You keep saying cells, because they will not
exactly.
get to the organization of an organ.
I think we can make cells and tissues. Organs, they need to develop even further, but there is a way to make organs, because there are a lot of companies that are decellularizing organs. So taking organs from a pig, decellularizing completely, and just staying with the scaffold, but they don't have a good source of repopulating these. We will be that source, and that's what we're going to do.
Thank you.
Yeah.
One more question. The FDA today is pushing quite significantly to reduce animal models and cannabis alternative. Are you considering also
Yes.
for screening methods?
Yes, I love these questions, because that's something that we're also want to promote. We want to, and that's one thing. I worked a lot with pigs and mice, and I love that we don't use, even we don't need samples, basically. We just need fibroblasts or something like that. We don't need oocytes, we don't need sperms, and that's a platform that can definitely promote less animal use. And also, a lot of the time, the translation between mouse, rats, and humans doesn't work, and that's solvable with this platform.
Thank you very much.
Thank you.